DNA is an amazing information storage molecule. Compacted into an incredibly tight space in our cells, we have this digitally encoded information using a four-base nucleotide system. But what I think is really cool about it is not just the information storage capacity, but how it fits in such a tiny space. Human cells have in a diploid genome approximately 2 meters worth of DNA in every cell. That's a huge amount compacted into a nucleus that's measured in micrometers. So we're talking about incredible amount of compaction here to get all of that information into those cells. Not only does it have to be compacted, but it has to be able to be accessed to transcribe the DNA into RNA, to replicate the DNA, and to segregate the chromosomes following replication during mitosis. And so all of these things have to take place without the DNA getting too tangled. In fact, this tangling concern was even recognized when the original structure of DNA was proposed by Watson and Crick. So even they recognized that there had to be some way to resolve this issue. In fact, they were using, whether they thought of it this way or not, a form of design triangulation and identifying that there would be a solution available to cells since their structure indicated that would be required in order for cells to survive. So how is it that cells overcome this challenge of an extremely long molecule of DNA that has to be opened up at times and has to be separated from the various other DNA molecules in the cell? Turns out we have an entire family of enzymes known as DNA topoisomerases. And the DNA topoisomerases, it's their job essentially to keep your DNA from getting too tangled. They do this by creating temporary single or double-stranded breaks by which they can alleviate things like supercoiling, which results from transcription and replication. They can also detangle knots and interlinks between molecules of DNA. And so these processes are carried out by this amazing family of enzymes, these molecular machines. I want to tell you the story of one of those molecular machines that we call topoisomerase 2 and its amazing mechanism by which it creates temporary double-stranded breaks, opening up the DNA double helix, passing another double helix through the break, and then putting that DNA back together. We're going to talk about the structure and the organization. We're going to talk about the regulation of how these enzymes are controlled and how they determine when and where to bind in the nucleus of a cell. It's really a fascinating story, and I look forward to sharing it with you. My name is Joe Deweese. I'm professor of biochemistry at Freed-Hardeman University in Henderson, Tennessee, and I look forward to spending some time with you on Wednesday, June 12th, to talk about DNA topoisomerases and the amazing things that we learn about the design of life through these fascinating molecular machines. Looking forward to it.